introduce. All right. Thank you, everyone, for coming to the bioinformatics seminar this week. Today we have Professor Shen from Texas A&M. I have known him for quite some time through our protein docking experiments. There is a community called Capri, which evaluates protein docking performance on specific many targets. And then Shen is one of the key players in the field. He always bring up very mathematical and CS-oriented approaches. So I can see that he's always trying to broaden the area of protein docking. That always fascinates me. So Dr. Shen is currently assistant professor in Texas A&M in ECE, and he got his bachelor's degree in China in University of Science and Technology in China. Then he got PhD in systems engineering from Boston University, and then did a postdoc at MIT under the supervision of by Professor Bruce Tidor. And he did research assistant professor at Toyota Technological Institute at Chicago. And now he's at Texas A&M. And recently he got two big awards funding. One is Mira Award from NIH, and also the Career Award from NSF. Those are the two biggest awards assistant professor can get. So he got two of them. This is really a great achievement. So today we will hear from his new developments. I'm very curious about which directions Professor Shen is going. The title of his talk is Forward Prediction and Inverse Design for Molecular Discovery. Okay, Professor Shen, then please start.
Thank you very much, Professor Kihara, for this invitation and generous introduction. Indeed, I think when we first met, I was a PhD student, and that was probably many years ago. It's great that to have this opportunity, because I'm aware of the great research and education at CS, at Purdue, including bioinformatics. So it's a great pleasure to be here and get some feedback from you about some of our recent work. So today we're focusing on the forward prediction and inverse design for molecular discovery. So I came from an engineering background, and biology was always something I was curious about, but not sure how I approach it. But one thing I think what engineering can really empower biology discovery is this concept of systems. Really, I mean, if you look at man-made systems as well as biological systems, you can regard both of them as hierarchical systems made of parts that work together for devices that work together for systems and so on. So it's with this perspective, I started to look at bioinformatics topics, and one of the focus of our research is by developing algorithms, we want to sort of reverse engineer the bioparts interactions or molecular interactions in the systems. And in the meantime, we want to design molecular interactions or introduce new interactions that will modulate the system's behavior. So that's our perspective, that by modeling and designing molecular interactions, we are able to enable molecular discovery for the biological systems. So today's topic include both aspects. First, we want to talk about the forward problem of molecular discovery, where you start with an input that is a cause and you predict the effects or the outputs. One example is that you can start with a protein sequence and it predict its structure. So this is a protein folding problem that Professor Kihara has contributed a lot and did very well in CUSP. And another kind of interaction that we are trying to focus here is, start with a molecular compound, a small compound or a drug. And you're curious about how it might interact with a target protein in our body. So that's a forward prediction problem. Another equally interesting and a little bit more challenging problem is the inverse problem that actually you want to start with the desired molecular interaction and design accordingly the drugs or compounds that can target the protein specifically, for instance. And this problem, inverse design, is usually ill-posed. So there are a lot of challenges with these issues. So in today's talk, I will mostly focus on the discriminative model for the forward prediction problem. The question is, how do we quickly predict protein-ligand interactions at the genome scale with interpretability? So that takes about 30 minutes. And in the last 10-ish minutes, I will talk about the inverse problem that we're developing a generative model to design combinations of compounds or ligands that can specifically target a disease module, not a single protein anymore. So let me start with the forward prediction problem. And as we know that drug design is no longer the one disease, one target, one drug scenario anymore, we're recognizing that our body is a system of many interacting molecules. And when we are designing a drug, we actually have to consider its effect on many, many bioparts in our bodies. So for this reason, when we are developing a drug, when we are searching in a small molecule space, which can go up to 10 to 60, we have to consider all its possible interactions with more than millions of proteoforms in our bodies. In the meantime, for each protein compound pair, you have to consider all kinds of measurements that might be needed, pharmacokinetic, dynamic, and so on. So this is a daunting combination for experiments that presents a need for computational prediction. So that's what our first topic is about. So I want to briefly talk about when we started this topic in 2017-18, what we were focusing on first, right? So I came from the principle-driven docking field. So one of the compound protein interaction prediction method is based on protein ligand docking. This is a very good at predicting the affinity based on the energy calculation. It's very interpretable. You can decompose energies into pairwise atomic interactions. However, you are solving a non-convex free energy minimization problem in a very high dimensional space. And not to mention that, sometimes you have to start with not a protein structure, but a protein sequence. So if you consider the gap between the structurally determined proteins and the sequenced proteins, and that gap is increasing. So that's the limitation of the docking approach. In contrast, we saw machine learning coming up that just start with the protein sequence. So the pros and cons are just like opposite to the physics-based, physics-driven docking methods. So they were not that accurate as when we started. They were just classifying interactions into binary outcomes back then. And then they were not interpretable at all. I mean, you don't know why a machine learning model makes that prediction, but machine learning is very, very fast compared to non-convex optimization. And then you can have very abundant sequences compared to very limited protein structures. So the pros and cons got swapped. So when we started this topic, we were asking, can we get the best of both, right? Can we make this realistic setting that we can predict the quantitative level, like affinity, not classification, and we only use sequence data so that it's broadly applicable at the genome level? We want to have a high throughput model that can enable the chemical proteomic combinatorial need. And at the same time, we want this model to have really interpretability or explainability. So these were the reasons we worked on this problem. And as a background, when we first worked on this problem, we introduced this method called DeepAffinity. So what it does is essentially embed the molecules first and then predict the affinity later, and then this whole process is trained end to end. I think it was our 2018 work. I think now a lot of people are actually working at affinity prediction and more or less adopting this similar framework, but let me introduce this as a background nevertheless, that we start with a pair of protein and ligand. Protein is represented as amino acid sequence or structurally annotated sequences. This structure annotation was predicted from sequences because predicting structures completely from sequences is difficult, but predicting whether it's solvent exposable or whether it's alpha helix or beta strand or any kind of secondary structure is relatively easy. For the small compound, we have SMILES or the chemical graphs. So we first have to embed these molecules. We used seq-to-seq back then because we were using the sequences for both molecules. So we could actually use a massive amount of unlabeled data. We can have more than millions of proteins in UniRef or chemicals in Stitch or PubChem that can pre-train the sequence-to-sequence autoencoder. Once this autoencoder is trained, you can use the learned embedder and concatenate it, feed it to the downstream affinity predictor. And then this can be trained based on just, we have 10 to 5 hundreds of thousands of labeled protein ligand pair. So this is one thing we did particularly was introducing an attention mechanism that basically put weights on pairs of compound atoms and protein residues that basically tell when we make that affinity prediction which pairs of atoms and residues were paid attention a lot, and we try to make that model interpretable in this way. So a quick summary of the results. If you look at the test set, compared to shallow models, deep learning does have lower root mean squared error and a higher Spearman's-Pearson's correlation. But we also paid more attention to these additional generalization sets. We actually completely excluded estrogen receptors, ion channels, GPCRs, tyrosine kinases, which are major drug targets for the following question, okay? So because if you don't have knowledge about a new class of protein targets, the performance for either shallow or deep models can be really bad. So if that's the case, what can we do? So the higher the worse for the 4 categories, okay? For the 4 categories of protein drug targets. And shown in the blue dashed line.
Hi, Yan. I think your microphone is a bit away from you. Can you adjust microphone position?
Okay.
Oh yeah, that's perfect.
Okay, I can sit a little closer.
Oh, okay.
Okay, it's a laptop. Okay, sorry about that.
No,
please let me know during the presentation. Thank you very much. So we now look at the root mean squared error of the blue dashed line, that's the shallow model random forest, and the red dashed line, that's the deep learning model. They are equally bad for the different cases. And we are asking this question. As we have few labeled data gradually revealed for this new class of drug targets, which strategy is better? Training a shallow model exclusively just for that protein target, or start with a meta model, meta deep learning model trained without the target class, and then gradually transfer it to the target. So let's see which one works better. As you can see, as you move from left to right, you're gradually adding more and more labeled data for the new drug targets classes. And you can see that actually the deep learning model is much better than the shallow model. So this is useful knowledge when we are questioning sometimes that deep learning is data hungry. What do you do when there's no or few data? So I think we answered both questions through the unsupervised pre-training as well as the transfer learning. One focus on interpretability was examined here by showing the marginalized attention on proteins. You can see that this is a compound in gray, and the colors were basically the marginalized attentions. So you see that around the binding site is a hotter color. So now, based on this background, we are going to introduce two recent developments. The first one is interpretability, or more, sometimes it's used interchangeably as explainability, but I think we are talking more about explainability, okay? So can you explain how your machine learning model makes such predictions? Okay, so we actually have to rigorously define and then assess the model interpretability first. So how do we define, interpretability for affinity predictor? I think it needs to conform to the scientific knowledge, human understanding, and the drug discovery needs. And as we know that affinity is usually decomposed as pairwise atomic interactions, and such interactions are very precious in the co-crystal structures and enables a lot of lead optimization in drug discovery. So we decided to define the model interpretability as ability to explain predicted affinities through intermolecular atomic contacts, in short, contacts. Unlike a lot of interpretability questions in computer vision, in our case, ground truth does exist and is expensive but available, okay? So the second question is, how do you assess model interpretability? So we defined the model interpretability assessment as the accuracy of the contact prediction, underlying affinity prediction. So we focus on this word underlying. So if a model just predict both affinity and contacts without contacts forming the basis for affinity prediction, then that's not intrinsically interpretable either. And we can use AURC or AURPRC for this binary classification as a starter. So let's first introduce a data set, okay, which includes both, which are labeled with both affinity and contacts, okay, and then see how the current attention-based methods work, okay. So let's look at the root mean squared error. That's the affinity prediction. The lower the better. So we introduced the different colors of different models for embedding proteins or embedding the ligands. And we are asking these different clusters like training, test set, new protein, new compound, and both new, okay? What you can see is that, as the interpretable models are good with the affinity prediction with a relatively low root mean squared error in affinity, their contact prediction accuracy is merely better than random, a little bit better than random, considering that contacts are minorities, okay? So the AUPRC is more suitable than AURC, and it's 0.004 for random. So that shows that current attention mechanism alone is inadequate. So the next question is then, how do you enhance the model interpretability, right? So we came up a few methods. First, I think it's impossible to let the model automatically learn where to pay attention. You have to teach the models with examples where to pay attention. So in this case, we introduce additional term in the loss to supervise attentions. So that's relatively simple. The second one is that you have to inject domain knowledge into attentions, right? So you have to teach the models with knowledge where to pay attention. So what we brought in as knowledge is a structure context awareness. As I said, this structure context is relatively easy to predict from sequence than predicting a structure. So we know that contexts are sparse, so we can use lasso to regularize them. We know that sequentially close residues should have relatively similar contact patterns to the ligand. So we use the fused lasso. We also know that spatially close residues should have a relatively similar contact pattern with the ligand. So we use the group lasso. So these are the additional regularization terms. And lastly, we also want to build this explainability intrinsically into the neural network architecture. So what we did was like we have a 3-stage gradual focusing. So we start with a protein sequence and a molecular graph. And at the beginning, we predict the solvent exposure as initial regularization. So we'll pay attention first to the predicted surface residues. And then we have the predicted attentions now is, now we have the predicted chimer binding residues built based on our joint attentions. And then these earlier attentions will regularize the later attentions. So we gradually shifting our attentions from surface to potential binding, predicted surface to potential binding chimers to potential binding residues and so on. This whole process is trained end to end. What about these yellow blocks, which are the relation neural networks? What we do is that we build those relational features like hydrogen bonding, halogen bonding, van der Waals interaction features, and then make them interact rather than following the current semi-empirical forms. So we use the neural network to learn how these features should be correlating with each other. So these are the how we enhance model interpretability. So we thus introduced the DeepAffinity Plus, that's taking the first 2 contributions, and the deep relations taking all the 3 contributions. So let's see how they work. So if you now look at the different models, again, we are looking at both affinity prediction, the lower the better, and contact prediction, the higher the better, AUPRC. So the 2 methods that stand out is the blue color, that is a deep affinity for, again, training, test set, new protein, new compound, or both new. And you can see that the root mean squared error for affinity prediction is as low as before. In the meantime, if you look
at the your microphone is off again. Can you get closer? Okay, I cannot hear you.
No, we cannot hear you yet. Yep.
Okay. We cannot hear you yet. I can see the mouse is moving. Okay. Oh, all right. So I saw the microphone noise from the microphone.
Can you hear me now?
Yes.
Okay, so I just remuted and unmuted me again. I turned on my video just in case that the bandwidth was causing the problem. I really apologize for
no problem.
For the audio quality. But now we are assessing the new models against the different sets. Again, with both the accuracy of affinity prediction and interpretability. You see that the accuracy level was like before, but the blue color deep affinity plus and the green color deep relations now has much better interpretability as before, like 10 times compared to before. The baseline was here. So this is the conclusion. If you look at the pattern of our predicted context, so let's first look at the baseline. Actually, there was no baseline. We had to retrain a attention-based binary predictor and modify it as affinity predictor. So this is Gauge Satchel from IBM. You can see that here I again show the protein in wheat cartoon, the ligand in green sticks, and then you see these dashed lines. So the gray dashed lines means the incorrect top 10 predicted contacts, and the red is correct. So there's no correct predictions here. And this is deep relations. And you can see that we have maybe 50% of the predictions correct. But more importantly, compared to before, you see that now these predicted contexts are very much focused in the binding side. I think that's largely thanks to the regularization of attentions. And then there are some origins of false positives. That's just binding not to the correct atom, but to its neighbor. Okay, so now we have this model that is explainable. We ask, how does this help you? We showed in other results that explainability actually can improve the modeling generalizability to new proteins and new compounds. In the meantime, we show that in various of applications, for instance, predicting binding site, again, this blue deep relations, deep affinity plus, and the green deep affinity, green deep relations work better than the baseline methods in binding site prediction. And using the binding site prediction, you can actually guide the protein-ligand docking to have much better or lower root mean square deviation. And we also used the relations for structure activity research. So, structure activity relationship study. So for this ligand scoring or screening question, we compared to the community methods that are structure-based. So it's interesting that compared to those structure-based methods, we beat more than 2/3 structure-based methods in the JAK2 sub-challenge. By the way, it's post-hoc. And we also, interestingly, in this case, the structure-free methods worked better than all the community structure-based methods. So I don't think they can replace the structure-based methods. When structure is available, you can use them. But I think this is a great indication of interpretability actually doing something useful. And you can use this interpretability for lead optimization as well. I won't show due to the time limit. So then the second direction we're following recently for deep affinity is actually, it's interesting thing. I have all these naive views sometimes, and sometimes they turn into curiosities and research topics. We're thinking that what are proteins is really a fantastic enabler for computer science research, actually. Think about a physical molecule, a protein. That is a text. It's a one-dimensional sequence written in an alphabet of 20 amino acids. It's also an image because it falls into a structure you can regard it as a 3D atomic cloud, 2D residue contact map, or distance map. And you can represent it as a graph as well. So it's really fantastic that you have this one physical entity that exists as model, as data in multi-modalities. So this is fantastic. Think about what is happening now in deep learning or artificial intelligence. We have NLP models good at learning the text. We have the CV models good at learning images, and we have graph models good at learning graphs. And it's really a fantastic opportunity for use-inspired new AI research. So the direction we are looking at is mostly focused on graphs. So basically we start with a 1D protein sequence and predict the 2D graph. You may ask, well, if both are from sequences, what's new? Well, when you are predicting the 2D graph, you not only use the protein sequence, you also use its family's protein sequences. So you actually consider additional evolutionary information here. So you can embed these proteins separately through a sequence embedder as well as a graph embedder, and then concatenate them. That's a very simple cross-modality protein embedding method that completely disregard the dependency across modalities, and these two embedding processes are separate. And instead, you can also let the embedding process affect each other. So we here introduce a very simple cross interaction model where you see that the embeddings of the graph and the sequence actually interact with each other through this learnable weight matrices. And this whole process is called a cross-interaction, cross-modality protein embedding. So let's see how they work. So again, let's look at a few methods like Gao, etc., or our own DeepAffinity Plus. And then let's look at if we use a single modality like a 1D sequence, okay, or a 2D graph. Here we also included the true 2D graphs as just a upper bound sort of, because they are not always available, but we'd like to have a check on them. So you can see that for the same protein, okay, whether both protein and compounds were same, but the combination is new, or the same protein is now in pair with a new compound, we are listing its root mean squared error and the Pearson's R. And then we summarize the performance in the average rank, the lower the better, or the Z-score, the higher the better. So actually for affinity prediction, using just the 2D modality actually does not necessarily work better than just 1D sequence modalities for this simple task of affinity prediction, especially for proteins you have already seen. But for proteins that you have never seen before, you see slightly difference, but still using just 1D sequence is fairly enough for even unseen proteins if you are just interested in affinity prediction. And then if you consider the cross-modality interactions or cross-modality embedding, now you see that even for this very simple task of affinity prediction, okay, this cross-modality embedding actually improves the results for the proteins that you have never seen before. So this is very interesting. And then we are also interested in affinity prediction, okay? In this case, I will show the whole table, and you will see that actually the single modality that's more important for affinity, for contact prediction is the residue-residue contact graph for the protein, the 2D modality. The best results come from the true 2D graphs, without very surprising. And again, in these cases, you see that the cross-modality model actually has even better AUPRC for unseen proteins compared to the true 2D graphs. Here we combined just the 1D sequence and the sequence predicted 2D graphs. They actually even comparable and even work better to the true 2D graphs for unseen proteins. I think these are very interesting results. And then beyond cross-modality embedding, we also have to think about another thing that is a self-supervision that has been known to inform better representation learning. I think this audience probably knows much better than I about the self-supervision, for instance, in text. Our 626 is used, is a very rudimentary version you can think of, but Transformers is using self-supervision a lot. You can see from this slide from Jan LeCun. And self-supervision is also introduced through all kinds of pretext tasks in computer vision. But our focus is now in graph. Remember, one is a representation of protein is a graph. So when we started late 2018 and early 2019, there were relatively few studies for self-supervision for graphs. So that was our focus. And then in one of our studies, we introduced, we systematically looked into the problem and asked the following 3 questions. The first question is, what's the best way to incorporate self-supervision in graph neural networks? I think when we started one method, one of the few papers was proposing self-training. So that's keep using the same labels, but predicted labels for the iterative process. We also look at the pre-training and fine-tuning versus multitask learning. The second question is, how do you design self-supervision tasks to improve the generalizability for graph representation learning? So we introduced several tasks, including the topology-based graph partitioning and the node feature-based clustering, as well as graph completion. It's like the image in painting. And then lastly, we ask, once you combine the proper self-supervision tasks and incorporate them into the graph neural networks properly, what do you gain beyond the generalizability? We found that it actually boosts the robustness of the models against the adversarial attacks as well. So we would like to test these ideas in our problem, because I think bioinformatics topics might be more challenging than the benchmark data sets we tested in this paper. So we were asking, well, can we, again, start with the very simple hierarchical RNN? Okay, it's not transformer yet, but let's introduce these self-supervision for text, like what they do in the transformers. And then more importantly, in the graph representation learning, let's introduce the self-supervision. Let's introduce the graph completion as the self-supervision task, and then separately embed these two modalities, and then maybe do the cross interactions as we did before. So we again look at the cross interaction model and look at what if we have no pre-training. So these are the results that you have seen before for proteins that I have already seen, or the proteins I have never seen before. I think you can skip the AURC or AUPRC and focus on the rank or z-score for a summary. So as you can see that compared to no pre-training, if we have the masked language model for sequence pre-training, or the graph completion for the graph pre-training, or doing both, we can actually improve the results for contact prediction for unseen proteins. Again, further improve the results. So this is some recent results that we very preliminary, we hope to further improve. So we also looked at graph contrastive learning with augmentations. Due to the time limit, I wouldn't talk more about it, but we are also very interested in how to apply this method to our probably more challenging bioinformatics problems. Okay, so now I have talked about the first topic of forward prediction. Here we are talking about how do you make interpretable prediction of protein compound interactions. So it's focused on the forward problem. So now let's look at the inverse problem, right? So one of the first questions you can ask is that, why do I have to design a new method for the inverse problem? Why don't I just use DeepAffinity and apply it to all possible ligands, and then see which one has the best affinity to the target protein, and then that's done, right? So the problem is that this is finding a needle in the haystack. Deep learning is probably making that process faster. It's like a magnet that can help you find the needle faster, but it's still very slow. So what if you can generate the perfect needles using the generative models that can model the joint distributions or the conditional distributions of identity and property? So that's our focus. I think this is already something that people have already looked at for drug discovery. And another thing we have to think about is that we are not just thinking about designing a drug for one molecule, one protein. We actually have to design a drug that works for a disease. And you have to consider that a disease module consists of subnetworks of protein-protein interactions. So these are the two things that we have to address compared to the first topic. And in particular, we are asking, how do you directly design drug combinations? Why we are interested in drug combinations? Well, this is because we know that actually drug resistance is actually a big issue for antimicrobial and anti-cancer therapies. So whenever you introduce a drug into our body, it presents a selective pressure to the tumor or the virus, and then they will try to escape through mutations or other mechanisms. So a well-established systems pharmacology approach to overcome the issue is drug combinations. So that's why we focused on drug combinations. So what are the challenges? Well, designing a single drug is already challenging. And if you consider the combinatorial challenge that the design space is much larger, that's an issue. But more importantly, the design principle is also challenging. So you have to actually consider what is the desired property at the systems level, okay? So there were no generative model for drug combinations. So we decided to explore the topic and see how it might work. So to address the first design space challenge, we introduced the reinforcement learning generative model. And for the conceptual challenge of design principle, we introduce a network biology principle as a reward in the reinforcement learning. So this whole process starts with embedding a disease through hierarchical variance graph autoencoders. And then given the disease embedding, we're going to design drug combination as graph sets through reinforcement learning agent. And this agent will also query for each generated compound whether it might interact with a protein in the disease module or not. This query is based on our previous DeepAffinity. And the biological networks will actually affect, again, this whole process. Let's start with how do you represent a disease? So the previous methods focus on just using the associated genes, and they ignore a lot of information such as gene-gene, disease-disease association and the biological networks. They also consider genes contribute equally to a disease. And we decide to think beyond that. And how do we do that? We decide to basically have two variational graph autoencoders to embed the disease, the gene-gene network and the disease-disease network. And then connecting through them is the sparse connections introduced by the gene-disease associations. And then this is done by an end-to-end joint training. And this hierarchy is modeled through attentional pooling so that genes can contribute differently to a disease. So let's first look at, and then for given disease embedding, you introduce a reinforcement learning agent that works in 3 stages using different rewards. In the first 2 stages, the rewards are disease non-specific. It's focusing on the chemical validity as well as the network principle. And in the last case, we actually make it disease specific. So I will skip the details about reinforcement learning. This is very similar to Leskovic's work. What we introduced additionally is that we introduced the adversarial loss to make sure that these generated drugs can mimic the known drugs or in the zinc databases. And we use the generalized Slice-Wasserstein to overcome the high dimensional challenges. And in addition, we ask, what can you do for the design principle in speaking of the network principle? So one work by Barbasi group in 2019 actually looked at the targets of drug 1 and drug 2 in a combination therapy and asked how they can potentially jointly cover the disease module. And they found that, if you look at the 6 different patterns, the one with the least side effect and the most clinical efficacy was this complementary exposure. So it's like the drug 1 and drug 2 separately cover different portions of the disease module as much as possible. So that's called complementary exposure. And therefore, we introduced this network-based reward, and we did some work to make sure that it can be calculated online and differentiable for the training. And you can introduce additional reward as well. We ignored toxicity on purpose, and we'll tell you why we do that. And first, let's look at the disease embedding. So we compared our disease embedding to different methods like Node2Vec, DeepWalk, and the HVGAE, and look at the different performance assessment metrics. And then we also look at the different ablated versions of our hierarchical variational graph autoencoder. So what if these 2 autoencoders are trained separately without interactions? What if they have interactions but without attentional pooling? And what if we have everything together? So indeed, you see that all the components contribute to the improved performance compared to the baselines or SODAs. The second part is about, what about the designed pairs? So we found that the designed pairs actually follow the network principle. We don't have many ways to examine that. We have to ask what's a portion of A minus B or B minus A, and then and calculate the network-based score. And we can only compare that to the existing drugs. But a better comparison would be, let's look at 4 diseases. For instance, we design drug combinations specifically for 4 diseases and first look at their network score. So there's no baseline methods. So we have to compare with 3 artificially constructed baseline. One is the gray one is the random pairs of drug bank compounds. So you take drugs and then take random pairs. The blue one is the drug combinations that are approved already by FDA. And the green ones are the random pairs of drugs for the specific diseases, but their combinations were not necessarily approved. So you can see that based on the network score, the red color of ours actually is more shifted to the right. So it's respecting the network principle more. In the meantime, we predicted the toxicity. So the more to the left, the lower. So you can see that those compounds designed by us respect the network principle and are also predicted to be less toxic. And lastly, we can ask, what are the mechanisms of the designed pairs? We found that they actually more or less reproduce a big portion of them at the gene level or at the pathway level, more or less reproduce the FDA-approved polypharmacology strategies. And sometimes there are some potentially new strategies as well, but actually we checked this later on. This dual inhibition was already known as well for breast cancer. So that's already validated. So with this, I would conclude my talk. And so we introduced a discriminant model for forward problem, and we focused on 2 recent development. One is interpretability or explainability. One is a cross-modality representation learning and self-supervision for protein data. And I want to emphasize that our forward prediction model can generate mechanistic hypothesis at the molecular level about how proteins and ligand might interact. And these hypotheses can be tested experimentally. For the inverse problem, we introduced the generative model. Again, the network principle can be specifically introduced as a reward to our reinforcement learning agent, and the corresponding designs can directly test the mechanistic hypothesis experimentally. So with this, I would conclude my talk, and thank you everyone for this great opportunity. I look forward to your feedbacks and discussions. I'd like to thank my students who make all this possible, and my collaborators who, including the computational and experimental collaborators who motivated a lot of these research topics, and our funding agencies as well. And again, thank you all for your attention, and I'm open to any questions.
Okay, thank you very much. Well, there are lots of new developments. It's very exciting. Any questions from the audience? Okay, I can start from my question. So in the first part of your talk, you showed it's surprising that actually the affinity prediction can also predict the binding residues by atom interactions. So in that case, so you didn't train. So it's not you only trained network based on the affinity prediction, but did you train both affinity and contact prediction simultaneously?
Right. So when you supervise attentions, you have to use the contact information. So that's part of the loss as well. So we tried without that information. You see that in DeepAffinity, it's just a little bit over the random.
Okay.
And then if you use regularization and the architecture, it will further improve. So it will be better than random, but just not at the level useful for drug discovery.
Sure.
So you definitely need to supervise.
Okay. And another related question is that, so when you compare the affinity prediction, you compared with several other like machine learning based methods, but if you compare with very conventional methods like Autodoc Venus score or I don't know Glide or those conventional drug screening tools, how does the deep affinity compare against?
Yeah, first of all, in our benchmark studies, we didn't compare to any of those conventional methods because they need protein structures as inputs and we don't need.
Right.
So
Okay.
it won't be a fair comparison. That being said, if you remember in the structure activity research as our relationship, we have two, uh, uh, D3R challenges, right? So in those cases, actually,
Yes.
the community participants are using autodoc vena scores or,
Right.
there are combinations as features. So they all use structures, and as a post-hoc assessment, in one challenge we beat 2/3, and in another challenge we beat the others. So I don't think we are going to replace them because whenever you have a structure, why don't you use them? This one is more like when we focus on when you don't have a structure, what's the best you can do?
Sure. Okay. Yeah, thank you.
Thanks, Yang. That was a great talk. So I have a
Thank you.
question, and maybe I missed this. So, what is the search space of the generative model in terms of like the drug combination that you're looking at? And would that be a search space that we could do a brute force search on it?
Right. So basically, the search space is the combination of k random graphs, and then the reward will make sure that these graphs are chemically valid, and then drug-like, and then respect the network principle. So that's this slide is related to the question you are asking. So iteratively, we are generating and completing this graph, and every time you add a link to the current state or the current intermediate graphs.
Okay.
And any bias or constraints for the state space can be introduced as here as well. Currently, just the random graphs and everything else is left to the reward.
Okay. Thank you. And maybe one more thing, and then this is just a very general question. Where or what measures we should meet as a computational crowd to say that we have a method that's good enough for discovery rather than trying to validate something that's out there?
So what we focus, so first of all, drug discovery is a huge topic. And I think, there are a lot of hypes from AI. And I think still they are looking at low-hanging fruits. So the real barriers and the most bottlenecks are probably not addressed at the moment. But I think gradually, with efforts, we can get there. But basically, if you stay in the chemical space, it's much easier. But whenever you move into the biological space, with a few data, a lot of unknown domain knowledge, and sometimes even undefined labels, that becomes very difficult. So now coming back to your question, in the small scope of the problems that we are looking at, what we focus on a lot is not just whether you can predict affinity, or whether you can generate a drug combination that's not toxic. It's more importantly is whether your models can generate hypotheses that can be experimentally valid tested. So if you think about biology, I mean, it's becoming more systems level study, more quantitative study in the past decades. And also thanks to all the great work in the field of bioinformatics and computational biology, especially those people who are working at the interface of computational and experimental work, we actually see that the computational methods are no longer just a post hoc validation of experimental results. They are actually able to help design experiments. So instead of asking what's in the experimental data, we can instead ask what experimental data you want to collect so that you can discover a knowledge. So I think that's what we focus on a lot. So in the molecular level, the contexts are giving you the experimentally tested hypothesis. At the systems level, what drug combinations are favorable, that reward is also a testable hypothesis, and our designs directly give you that testable hypothesis that can be experimentally validated. So that's the direction that we are trying to follow.
Great, thank you.
Thank you.
I have a questions. Yeah. So about the sequence embedding and graph neural network. So did you use the multiple sequence alignment informations?
I know that's a big thing in the recent cusp. We haven't. We are looking at it, but a lot of people are doing that as well, using transformers. I definitely have seen a MSA transformer recently.
So do you think that if we use the multiple sequence alignment for the input informations, the performance will be improved?
I think conceptually that should be the case because you include additional information from co-evolution. But in practice, we have to design neural network models, architectures that can bring out such information. So sometimes it's like the signal to noise. Sometimes you have more data, but that there are more noises in the data as well. So how to extract them? That's the fascinating part of doing bioinformatics research and use and develop algorithms for bioinformatics.
Thank you.
I don't know whether that answers your question.
Yeah, but performance is already good. So even you use the single sequence information, it's amazing.
I don't think it's that good yet, but thank you for the comment.
Okay. Any other questions? So I was really interested in the embedding of disease. So it's no, we have been I have been working more to in the 3D structures, so because it's concrete and it has interaction and so on. But now to go to systems level, you embed it. You have embedding of the disease, which explore which can broaden the research field significantly and has a potential practical impact here. So here the way you so I wanted to ask how you could improve or like extend the disease embedding. Actually, in this seminar series, a couple of weeks ago, I invited Shen from KAUST. Maybe you know him. And he in a couple of slides, he talked about placing the disease into the network to reveal the relationship or similarity and dissimilarity of diseases. And I don't remember the detail, but
Gotcha.
he used the clinical data of patients, and also even some written documents by medical doctors. And can you expand such different type of information to further enrich the disease embedding?
Right. So definitely. I think that's a great question, and definitely, there are a lot of things we can do. So one incremental thing that we can do is, as you said, for the disease interaction network that we constructed, we can use additional information to construct that single disease network. But it's also possible, as you said, that we can consider like multi-views of the disease interactions from all kinds of data, and then you would do some kind of cross-modality metric learning to better embed those diseases. So I think that's also the idea in contrastive learning as well. You're trying to figure out which one is closer, which one is more distant. So there's definitely a lot of room for that. In the meantime, you may want the basis elements, the basic elements entering the disease embedding to be more directly mechanistically relevant, probably. So we start with the genes because we think that is mechanistically explainable, but other things can be incorporated as well. I think there's a balance between the interpretability and the accuracy, and depends on the need. But there's a lot of rooms now in embedding the knowledge graphs. A lot of the knowledges in our biology exist in graph. So so that's a great topic that we hope to contribute more to.
Yeah, it's very interesting now using embedding. Many things can be represented compactly and can connect many things together, which are not possible before. Molecular level, atom level, or like a disease level or phenotype level. There's techniques to be able to do that. I'm not sure how well it works, but anyway, you can connect many things. That's really exciting.
Right. I think a lot of the studies are gradually revealing the possibilities, and even though they might be at the infant stage for now, but I think it's very exciting. It's there are a lot of work to be done in the theoretical, numerical, and application levels.
Yeah, I think so. I think so. I have been always thinking that how I can depart from structure level and the compound level and atom level to more systems level. So and when I thought about systems level, I also thought about pathways or lots of proteins in a cell or something like that. But this is way beyond. And so it's going beyond to the physiological level or disease level. So you can jump to any levels of the biological description.
Yeah, I think then it's probably time to again, rediscover the classical mesoscale systems modeling techniques and reincarnate them into deep learning maybe. Instead of trying to reinvent everything from scratch. So I think there are a lot of opportunities, and we're not doing that well compared to what we want to at the systems level. So this is just one of our early efforts, and we definitely hope that the molecular discoveries can be more informative of the system's behavior, and it can affect the system's behavior eventually to the phenotypes, including disease. Right.
Okay. Any other questions from audience? Okay. Let's thank the speaker, Professor Yan Shen, again. Thank you very much for a wonderful talk. It was very inspiring.
Thank you very much for the opportunity, and thank you very much for the discussion.
Okay. So thank you very much. So we will conclude this seminar today. Okay, so Yan, hopefully we see each other in physical settings sometimes.
Yeah.
Okay, thank you very much. And we have a seminar, this seminar next week too. So please join next week too. Thank you very much
for incorporating my schedule. Sorry that you had to.
Oh, no. It was really great to have you. I really wanted to have you.
Thank you. Thank you so much.
Thanks, Yang.
Great to see you again. Nice to meet you.
Nice to meet you.
Look forward to this interaction next time. Thank you.
Bye.
Bye.
Okay, thank you. Bye.